Okay, this is the Professor, CBMD PhD, and today's Thursday, November 23rd, 2023. It is 4:29 and 31 seconds, a.m. Eastern Standard Time, and today's research, lung cancer cells. So let's jump to it here. Lung cancer cells. Memory suggests new strategy for improving treatment. Date November 17th, 2023. Source: Memorial Sloan Kettering Cancer Center. Research shows that some lung cancer cells retain a memory of the healthy cell where they come from, one that might be exploited to make an emerging type of lung cancer treatment called KRAS inhibition more effective. The study looked specifically at lung adenocarcinoma, a type of non-small cell lung cancer that is the most common type of lung cancer in the U.S. and responsible for 10% of all cancer deaths. This cancer is frequently driven by mutations in the KRAS gene. As for a long time, cancer-driven KRAS proteins were considered undruggable within the last few years. However, the U.S. Food and Drug Administration approved the first KRAS inhibitors, with quite a few more in clinical trials, but they don't work for everyone, and most patients' cancers eventually acquire resistance to the drugs and come back. And to understand the MSK discovery and its implications, it is helpful to know a little lung biology. Within the lungs, oxygen is absorbed in carbon dioxide released via air sacs called alveoli. The lining of the alveoli is made of two distinct types of cells, alveolar type 1 and alveolar type 2. And while they are similarly named, these two cells couldn't be more different. Alveolar type 1 cells are long and thin with a large surface to facilitate gas exchange between the lungs and the bloodstream. Alveolar type 2 cells, meanwhile, play a caretaking role, secreting compounds that are important for the health and function of the lungs, as well as helping maintain and repair the lungs by dividing to create replacement alveolar type 1 cells. You can think of them as stem cells with a day job. The big problem comes when lung cancer cells, which typically develop from alveolar type 2 cells, take on some remembered properties of the alveolar type 1 cells that alveolar type 2 cells differentiate into when they are playing their stem cell role. Scientists call these cancer cells alveolar type 1-like cells. In healthy cells, KRAS plays a key role in regulating cell growth and division, but when the gene becomes mutated, it can lead to runaway cell proliferation. As KRAS inhibitors can switch off this explosive growth, greatly diminishing tumors, but they still leave behind pockets of cancer cells that aren't sensitive to the drug, and then also give the cancer a chance to develop new mutations to resist the drug's effects. The research team painstakingly studied these residual cancer cells to uncover the mechanisms of this resistance using genetically engineered mouse models, mice implanted with patient-derived tumors and tumor samples from patients. They discovered that the cancer cells that remained after treatment were these alveolar type 1-like cells. They also found these cells have the capacity to reignite the cancer's runaway growth. Importantly, they found that if you get rid of these alveolar type 1 cells, it greatly improves the treatment response to KRAS inhibitors. Eliminating those cells in experimental models is relatively easy, but doing so in the clinic will require further research. They actually live in a very exciting time with fantastic pharmacology that can engineer molecules to bind to a certain cell type and kill them. This is how CAR T-cell therapy and antibody drug conjugates work. Now that they've done these proof-of-concept experiments, the next step would be to find surface proteins that are unique to these alveolar type 1-like cells, and then develop a therapeutic that can bind to them and kill them. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Nature of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.